Hello folks, today we will be talking about buffers and pKa. Today's learning outcomes. We should be able to describe why buffers are good at resisting changes in pH. We should be able to predict if a molecule is protonated or deprotonated depending on its pKa and the environmental pH. And finally, we should be able to draw a titration curve for a polyprotic acid. So buffers, pKa and pH are all kind of review topics from previous classes, but I understand that these are also very challenging topics, so I always like to devote a day in class to this. But first, let's discuss today's Meet a Scientist moment. So this is Sarah Rankin, professor of leukocyte and stem cell biology at Imperial College London. And so this professor was diagnosed with dyslexia and dyspraxia later in life, so she is neurodivergent. Her whole life she kind of didn't understand why she was different, and then finally received these diagnoses and was able to kind of make sense of who she is. She studies the impact of bone marrow, stem cells, and leukocytes on disease. So how does your bone marrow respond when you get sick? Some pretty interesting stuff, folks. Now let's get to talking about buffers, pH, and pKa. And first, just a quick reminder that since we are working in water, we're in biochemistry, humans are made of water, we should remember that water itself can act as an acid or a base, so we'll keep that in mind. And I'll just demonstrate this quickly with this thought experiment below. So let's say we have a proton at point A. Here's our proton. And a base at point B. Here's our base. In aqueous solution, how does the proton get from point A to point B to react with the base? A note is that the proton appears to get to be faster than the rate of diffusion. So the reaction occurs faster than it would take from H, the proton, to move all the way to my OH, my hydroxide. The answer is already on the slide, folks. There's lots of water in between. So let's draw some water molecules in between. Here are some water molecules. You can see that they're performing some nice hydrogen bonding interactions between the lone pairs and the hydrogens, although that is going to make the slide a little bit confusing to look at, so I'm going to erase those and leave those off. But you can still be imagining the hydrogen bonding interactions in your head. So how does this reaction happen faster than it takes from that proton to move to the hydroxide? Well, all of the waters in between can participate in this acid-base reaction. So the base actually just grabs the grabs the proton from the first water molecule. So that water molecule can grab a proton from the next water molecule. That water molecule can grab a proton from the next water molecule. And finally, the last water molecule can grab that proton. So this slide, folks, just to remind you that water can act as an acid or a base. Since we're talking about acids, bases, and buffers, that's an important thing to make a note of. Let's move on to an idea that you've already learned, but let's just think about it again for a second. pH. pH tells us about how acidic or basic a solution is. If you're a math person, we definitely want to look at the equation. So pH equals the negative log of the concentration of protons. So when we're saying pH tells us how acidic or basic a solution is, pH really tells us about the concentration of protons in solution. Okay? Here on the screen, you can see this graph is showing you that when we have a greater concentration of protons, we're going to have a smaller pH or a more acidic pH. When we have a lower concentration of protons, and therefore a higher concentration of hydroxide in water, we're going to have a higher pH or a basic pH. Some vocabulary for us, just as a recap: we have acids, things that can donate protons; bases, things that can accept protons; and then conjugate acids and conjugate bases, depending on whether you started with a base or an acid. If a base gained a proton, we'd say that was its conjugate acid. If an acid lost a proton, we'd say that it was its conjugate base. And so that's pH. Quick recap. Now we also need to discuss pKa, but I want us to keep in mind, folks, pH and pKa, while they use the same scale, they are not the same thing. pKa has everything to do with how strong an acid or a base is. On the screen, you can see my pictures covering up this equation. Don't worry too much. We're not going to focus on the equation in this class. We're going to try to understand conceptually what pKa is. So the strength of an acid is defined by its dissociation constant, or Ka. So let's just take a look at this. Use the equation real quick, but just think about what's happening here. So if K is large, I'm telling you it's a strong acid. So if K is large over here, it means that the numerator is large and the denominator is small. That means that most of our acid has dissociated, has donated a proton to result in hydronium being formed. Again, we're in aqueous solutions. So a large Ka tells us that the acid is strong because it's donated most of its protons. Now, we don't normally think about Ka a lot. We normally think about pKa. Why? Well, probably because it's so similar to pH. They use the same scale, so it's easier to think about. So pKa is the negative log of Ka. So the smaller the pKa, the stronger the acid. The smaller the pKa, the more likely it will be to donate its proton. So to wrap up, folks, pKa tells us how likely an acid is to lose or give its proton. We can also think about how stable the conjugate base is. Things that have a very stable conjugate base tend to be strong acids. And so let's compare two common pKa's to kind of wrap up our understanding of pKa. So two important functional groups in biochemistry are carboxylic acids and primary amines. But carboxylic acids have a much smaller pKa, whereas primary amines have a much larger pKa. Well, that's because a carboxylic acid is going to be much more likely to donate its proton, because its conjugate base is incredibly stable. If I were to draw this out, folks, you would see the functional group looks like this. This is incredibly stable because we have resonance stability of the negative charge. So I could draw the resonance hybrid, but I'm not going to do that. On the other hand, this primary amine over here is weaker. Its pKa is 8 to 9. It's much less likely to give away its proton. It is more stable as NH3+. And so it will be less likely to donate its proton. So that is a wrap up of pKa. Now let's talk a little bit more specifically about pKa and buffering.